I'm host Bernal, and today I'm going to talk to you about our work on selective motion artifact reduction for improving perivascular space quantification in brain MRI. So let's get started. So perivascular spaces are tubular structures which appear hyperintense in T2-weighted scans, and they can be found in the basal ganglia and also in the central semivalley typically. And these PVS are thought to play a role in cerebral hemodynamics and also interstitial fluid exchange. And they are typically microscopic, but they have been seen in large and abundant in patients with some risk factors and some diseases. So we think that they can be a biomarker of brain health function. However, in order to assess whether this is the case or not, we need to quantify them accurately. And we've seen throughout the years that if we have a high quality scan, we can carry out segmentation quite nicely. But if the image has been corrupted by, let's say, motion, then segmentation results are suboptimal. And so to deal with this problem, we've proposed a three-step framework. First, we given an input image, we examine its quality. If it's of low quality, then we reduce motion artifacts and then segment perivascular spaces. But if it's of high quality, we go straight and segment perivascular spaces because filtering a high quality image is actually counterproductive. So let's talk about each one of the steps now. So first step, automatic image quality control. And so we use 4 quality metrics that evaluate motion and noise and that we computed using MRI-QC. So first, entropy focus criterion, which evaluates ghosting and motion artifacts. Then the foreground background energy ratio and the mean background intensity, which evaluate the signal in the background area. And also the signal to noise ratio. So after computing all these features for each one of the input images, we classify them according to a logistic regression model. Second step is motion artifact reduction. And so in this particular scenario, we proposed a protocol-specific method because we do not have access to the original case space and the entire processing needs to be done retrospectively. So what we did was to look at the case space of original images. And what we observed is that this case space we obtained via the Fourier transform exhibited some sawtooth patterns or pie-sliced patterns that are related to motion. So what we did to compensate for the motion problem was to nullify these segments that were affected by these motion artifacts by replacing them by zeros. And after that, we used the total variation optimization scheme and also the Fourier transformation to go back to the image space and to reconstruct the image and obtain an enhanced scan. So as you can see, and as pointed by the red arrows, motion artifacts are being reduced, and also some PVS that weren't evident before are now visible. The third step is PVS segmentation, and so in this particular case we stick to the 3D friendly filter, which is the gold standard for PVS segmentation. And we segmented PVS in 2 regions, the basal ganglia and central semivalley, and computed measurements afterwards, such as the count and the volume of them in the brain. We tested our proposal on data from an ongoing study called the MSS3. In this particular case, we had access to 60 patients. Imaging was carried out on a 3 Tesla scanner using a T2-weight sequence, and voxel resolution was more or less isotropic. All scans were visually rated using the Potter scale, which examines PVS burden in basal ganglia and central semivalley and classifies them in range between 0 and 4, being 0 the minimum and 4 the maximum PVS burden. And you can see for this particular sample, most values are concentrated around 2 and 3 for both the basal ganglia and central semivalley. And also an experienced image analyst examined the quality of these images, and we found that around 20% of the images in this sample were affected by motion artifacts, which is quite dramatic for this small sample. Alright, so let's talk about the results now. So we first evaluated the performance of our image quality classification scheme. And so we tested first the performance of classifiers using descriptors individually, but also together, all descriptors together. And we used the ASU-ROC as performance metric. You can see that when we use all descriptors in a single model, we obtain higher performance. And this 96 in our small sample means 2 misclassified cases out of the 60 that we had. So very good classification performance. What we did afterwards was to evaluate the polytheory correlations between visual scores and computational measurements that were obtained with the original images. That images that were selectively filtered using the total variation optimization scheme, and also our protocol-specific pipeline. And so as you can see, polytheory correlation values increased after using selective motion artifact reduction strategy. And this was particularly evident for the central semi-valley. We also detected some systematic errors segmentation methods make, and that may be research in the future. So first, here in this area, in the central semi-valley, we observed that long and thin PVS are often not segmented completely. They're typically fragmented into multiple pieces, and this affects counts of PVS in the central semi-valley. And the second effect is that sparse PVS are typically segmented better than those in proximity. And this has to do with the multi-scale approach in the French filter that tends to combine PVS that are near each other. In general, the segmentation in the basal ganglia was much better than that in the CSO, and it's because the motion artifacts are much more detrimental in this in the central semi-valley than in the basal ganglia. So just the final remarks. In this work, we proposed a framework which selectively reduces imaging artifacts, and we observed that this facilitates also PVS quantification, especially in motion corrupted images. And this is needed because the prevalence of imaging artifacts can be as high as 20%, as reported in some large-scale studies. Also, we observed that PVS segmentation is better when we filter selectively, but also when we use a protocol-specific pipeline. The problem with this type of approaches is that there is a lack of generalizability. However, coming up with multiple protocol-specific pipelines is not something feasible, and so in the future, it might be better to look at some artifact-robust PVS segmentation strategies instead. And so with that, I conclude my presentation, and I thank you very much for your attention. Thank you.